Good evening, everybody. Welcome along to what is the last Cafe Scientifique of the season. My name is Scott McIndoe, and I'm going to be introducing our speaker tonight. And I'm happy to announce we're going to go out with a bang because we have tonight John Burke, my colleague from the Department of Biochemistry and Microbiology. I'm going to give you a little bit of background on John. John hails from south of the border. He was born in Oregon, and he grew up there before going to the University of California in Berkeley to do his undergraduate. Then he went to UC San Diego for his PhD before heading over to the UK to the world-famous MRC lab in Cambridge, where he did his postdoc before coming here to Canada to join UVic as an assistant professor in 2014. He has since proved himself to be an absolute superstar and has advanced through the ranks incredibly quickly. He just got promoted to full professor this year, so he's done amazingly well. He has an incredible list of collaborations. He's collaborated with over 40 people around the world, so there's a lot of people who want a part of what John is putting out there in the world. So he's going to talk to you tonight about the future of drug discovery. So John, welcome to the Cafe Scientifique stage.
All right, thank you everybody. It's always exciting as a professor giving a microphone with no visual aids and just talking for an hour. So hopefully this will be an enjoyable story. So my name's John. I'm just going to say I'm happy to answer questions as the talk goes on. So if there is something that I say that isn't clear, feel free to raise your hand and ask a question. There will be time for question at the end, but I'm always happy to do questions in the middle as well. So today I want to talk to you a little bit about how drug discovery is currently done and how drug discovery is going to change. And so I think what's useful is maybe I'll give you a little bit of background about myself, my training. I'll talk a little bit about how drugs are currently developed in the pharmaceutical industry. And then I'm going to talk a little bit on how the work my lab is doing in collaboration with a huge number of different both pharmaceutical and academic partners can help. Can everyone hear me in the back? Yeah. Perfect. Okay, fantastic. So I began my training as a chemist. So like Scott, even though I'm in a biochemistry department, I did my undergraduate in synthetic organic chemistry. So really what I was starting my career with was trying to make molecules, and molecules that hopefully would have some effect in targeting of different diseases. I rapidly found that my fingers were completely all thumbs and that was not going to be my long-term career path. But as a beginning, that's kind of how I got into this. And so I had an initial orientation of looking into how academics and the pharmaceutical industry make molecules that could be useful. I went to my PhD at the University of California at San Diego, and I did it on a very esoteric problem. And that esoteric problem was, if you think about every cell in your body, a unifying feature of all of them is it has a big glump of fat surrounding it. Every cell essentially has this layer that surrounds it, and essentially what keeps your cell inside and prevents all of this contents from spilling out. What that means, though, is that there has to be a way that every cell in your body can respond to a signal that comes into it. Right? And so what happens is that there have to be molecules on the surface of that cell, and they have to respond to some extracellular signal. And every part of us essentially is developed by the correct orientation of every cell being able to respond to a signal. So what I did in my PhD was I developed a method using a technique called mass spectrometry. I'm not going to go into the details of this, but essentially it was a very esoteric problem, was how do the machines of our cells, the proteins, and so when I say protein, I mean the little molecular machines inside every cell of our body that do every task you can think of. They chew up food, they translate our DNA into other protein machines. How do those molecules actually come to that fat surface, right? And so what I did, and this was convincing my parents this was a good idea, was I took this molecule from cobra venom. It was a molecule we could make an incredible amount of this, and it's one of the best enzymes or proteins in the world that binds to kind of a fat surface. And so I became the world's expert at essentially trying to figure out how those molecular machines in our body bind to lipids. I remember when I gave my PhD defense that my father was very impressed, but he said, why does anyone care in the world why you would want to know how a random cobra venom protein comes to a fat surface? And I said, just you wait, this will be useful at some point. And he said, I'm not totally sure that's true. All right, so that's my background. So I essentially was someone who was studying the protein machines in our body. At that point I was studying the protein machines of a cobra venom. And so I went to Cambridge. And in Cambridge I said, what is the biggest biological challenge that I could probably go to? And so the system that I tried to apply that same technology towards was a set of molecules known as phosphoinositide 3 kinases. And that's a lot of what I'm going to talk about for the rest of it. I'll explain what that is a little bit later. But just for now, the simplest way to think about this is that if you look at every cancer that's known, most cancers are driven by mutations that occur in different proteins in the body. This enzyme just happens to be the second most frequently mutated protein in all of human cancer. So if you know someone who's had cancer, there's about a 20% chance that it's from a genetic mutation in that specific molecule. And so the way that molecule works is it also has to get recruited to a membrane surface. It has to go to that surface of a cell. And the reason it's so frequently involved in cancer is it essentially acts as a pro-growth signal when it goes to that cell surface. And so essentially my whole postdoc was applying that same set of technology to that problem and really trying to figure out how in disease, so when that protein becomes mutated, how does it actually become differentially regulated? So we'll go into the details of this later, but I think what I want to sell here a little bit is that my PhD really was on real general basic science. It was taking a simplified system, becoming really expert on a given technique, and then sometimes the actual major disease challenge pops into your hand and you have the right tools, but you develop the tools for the love of the science, right? And so I think that's an important thing, and we'll talk a little bit more on exactly how it works. So that's my background. And then I came to start my lab here at UVic. And so I think what might be useful, how many of you know roughly what the cost of drug development is right now? Anyone have a guess of what that number is? It's about 3 billion is the most recent estimate. And it's a little bit variable. So the lowest estimate you can find is for a new drug, it's about 600 million. For the high-end estimate, it's somewhere on the order of 5 billion. Does anyone know what it was in the year 2000? Take a guess. It was about I think it was 800 million was the estimate then. And if you look in 1990, it was about 50 million. So you can see it's an almost exponential, not quite exponential, but it's a non-linear growth. So it's getting more and more complicated. So essentially how drug discovery currently works is that you have this preclinical testing. So that's essentially the companies and academic partners, everyone's looking together and they say, what is the molecule that we actually want to look at? So what is some target? I've got some molecular machine in the body. It looks like it's misregulated in disease. I want to create some type of small molecule or some approach to hit this and turn it off. That's what takes up about 70% of the money. They finally get up the molecule that they want. They go through a tremendous amount of screening. Then once they have that molecule, then it has to go through animal testing. There's all of the complications that go through with animal testing. It has to go into safety trials in humans. That's kind of the small phase 1, if you've heard of phase 1 clinical trials. Then it has to go to phase 2 clinical trials, which is where you actually test for efficacy in a small subset of patients. And then you go to phase 3, where you expand that into the large set. So that is kind of the pipeline that's currently there. And the pipeline really can't function at the cost of what a drug is right now. So there have been some tremendous advances. I'm going to talk a little bit about some of the stuff that my lab does. I'm going to integrate. A lot of it will be work that my lab participates in with a wide group of partners, and I'll try and break in the new aspects of it that hopefully will bring that cost down, but this is kind of a large problem that's going forward. Okay, so that's my intro. So let me now go back to this PI3K pathway. So I'm going to try and break this down in a way that probably most of you at least have some reference to. Once again, going back to the idea of a cell and any cell in your body and how it receives signals. So we're going to talk about something signal transduction is the scientific way of saying it, but I think the simple example that everyone might know is, does anyone here know what insulin does in our body? Yeah, so insulin is the thing that regulates our blood sugar. Insulin essentially is something that your body secretes from the thyroid, and then it's not from your thyroid, sorry, from your pancreas. That was a good sign. From your pancreas. So insulin secreted from the pancreas, and essentially you have a sensor in your pancreas that's sensing the amount of glucose in your body. Now that insulin then gets secreted and it travels all throughout your bloodstream. Has anyone ever thought about what insulin actually does? Yeah, so insulin binds to cells that have insulin receptors. So this is that example of this signal transduction. So there's going to be cells in your liver, there's going to be cells all throughout, there's cells in your muscle, there's cells all over your body that can respond to that insulin. Now, what insulin actually activates in your cell is that molecule PI3 kinase. So PI3 kinase is actually the main effector molecule that comes into the cell. So you've got a cell, you've got insulin outside, you have this insulin receptor on the surface of the cell. That receptor can engage the insulin and then has a signal on the other side of the molecule that turns on this PI3 kinase. Now, I'm not going to go too far into what that PI3 kinase does. I could tell you lots of fun details about kinases and lipids and signals and 20 different proteins that it activates. But from the simple process, essentially think that like insulin binds, insulin receptor gets activated, it brings this molecule from the inside of a cell to the outside of a cell. That's the simple viewpoint of it. Now, the issue is, as I said, this PI3K protein is the second most frequently mutated gene in all of cancer. What happens in these mutations is that even in the absence of a molecule like insulin, that protein goes to the surface of a cell and it turns on pro-growth signals. So normally in a healthy tissue, you only have growth when you have a signal saying, turn this pathway on. In a diseased tissue, you're going to have, or in a cancer tissue, you essentially are going to get this uncontrolled growth at all times, right? And so that's the fundamental question my lab is trying to answer is A, how do these mutations cause this to come to the surface at all times? Are there ways that we can target this in a mechanism where we can stop that uncontrolled growth from happening specifically at that mutated form and not the wild type form? And is there a way that we can do this in an optimized way that we can speed up the development in preclinical development? Does that insulin story make sense for most people? Okay, so let's talk a little bit now about the initial drug discovery elements to target that enzyme. So when I say that this was identified as this major cancer target, that happened during the genetic kind of revolution. So in the year 2000, the whole human genome was sequenced. And then almost immediately what the major efforts of genetic sequencing were was you go and you look in different diseases and you say, here is this reference population, which is kind of the healthy average population. And we look in a variety of different disease states and we look for mutations that are different between healthy controls and the disease states, right? And this was, you know, many of us were here. I think almost everyone in the room was here when that was announced. There was definitely some overselling on the aspect that now that we know the genetic basis for every instruction in our body, we'll be able to figure out the problems in disease and we'll be able to solve these very quickly. Now, of course, that hasn't panned out as directly. There's been lots of advances that have come from it, but it hasn't been as easy as knowing, here's this mutation, let's immediately turn this off. So this PI3K enzyme in about the year 2004 was identified as this major mutated enzyme in cancer. And so immediately there was a gigantic investment by the pharmaceutical companies to develop molecules that could specifically turn this enzyme off. Can anyone think what might be a problem of turning the molecular machine that responds to insulin off? Might be. Doesn't actually kill you, but it's very toxic. It essentially acts like kind of a diabetes in a pill. Because what you're doing is, so right now, and many people are having dinner right now, your blood glucose is going up, right? As your blood glucose goes up, your pancreas responds by producing insulin. The insulin works, and this is the only aspect of the insulin signaling pathway that you need to know. Insulin binds, insulin receptor, this PI3K gets turned on, and the glucose transporters in that cell go to the surface. When the glucose transporters go, those are your sugar transporters. The sugar goes from your blood, and the sugar goes into the cell. So essentially, it's a regulated system. When you get blood sugar going up from eating, your blood sugar comes down from the response of this pathway. Now, what that requires then is that coordinated function of how insulin works with the sugar, with that receptor, with this enzyme. So, and this is also some of the diet link with cancer is that if you have overactive insulin signaling at all time, you're also overactivating this PI3K signaling pathway, and some of the connection in between type 2 diabetes and increased cancer levels is through this increased growth signaling. Okay, so pharmaceutical companies developed this first generation of compounds, and this compound turned off PI3K signaling everywhere in the body. And so this one I'm totally happy to take questions. I'm going to try and explain what happened here with these molecules, and if it doesn't make sense, I'm totally able to repeat it. But it's something that even with my students, I have to explain this a couple times. So they gave the drug, and you give the drug, essentially you block the signaling pathway from going. Those glucose transporters never go to the surface of the cell. So blood sugar just keeps accumulating, accumulating, and accumulating. Does anyone know what your pancreas does when your blood sugar keeps going up and up and up if you're insulin responsive? Makes more. Your insulin just goes through the roof, right? So every surface of the cell now, eventually, so you have this drug there, but you're just pumping insulin. And so these drugs went into clinical trials. If you took the blood of a patient who got one of these drugs, if you looked at their insulin levels, if that person didn't have the drug in their system, they had levels of insulin that would kill a person almost instantly, right? Because you got this incredibly increased dose of insulin going into the system. Because you had this drug turning the pathway off, it actually doesn't have those toxic effects of too much insulin. But what actually happens is you gave a drug to turn a molecule off. If you look at those patients, they actually had that pathway turned up higher because of that homeostatic effect of the insulin going at these hyperactivating levels. Does that make sense? Yeah, so this is the kind of thing that like when these genetic discoveries were made, they looked at this only from the perspective of a cancer cell. And they said, how do we turn this off in the cancer cell? You don't necessarily think about how your body is going to respond systematically to that effect. Okay, so where does my lab come in? Then we started thinking, we saw these results from clinical trials. There'd probably been at that phase about $10 billion of investment in the pharmaceutical industry in developing molecules targeting that pathway. And there'd been some successes. So there actually are some molecules that are approved for a specific class of cancers. But in terms of that promise of 20% of all cancers out there, it really didn't pay off in terms of everything coming through. So the real next goal was thinking about how do we actually then look at the disease state? So if you could make a molecule that only turned off the mutated version, but doesn't turn off that wild type version that actually handles all of the insulin responsiveness of your cell, you could still have your liver expressing those glucose transporters, bringing the blood sugar down, but the cancer cell, which depends on the mutant, then can't actually generate that pathway, and then the tumor cell dies, right? And that was the idea. And so maybe, actually one point I didn't make here. When I say this gene is highly mutated in cancer, it's what's called a somatic mutation. So a somatic mutation is a mutation that occurs at only one point in your body, right? So if you develop a tumor, you essentially have a subset of cells in your body that have a different set of genetic instructions, and that genetic instructions are telling them to grow uncontrollably, right? That's not something that's in every cell of your body. So there's a very sub-localized part of your tissue that essentially has that mutated form, and then that wild type form that is required to take down blood sugar to actually respond to insulin is everywhere else. So ideally what you want is you want to find something is, can we identify a difference in that mutant form from this wild type form that we can use? So now, how do you actually do those type of experiments? So now I'm going to tell a little bit about my actual work. I'm just going to keep an eye on time here. So here is a 3D printed molecule of what a protein actually looks like. When I say a protein, this is probably 500,000 times larger, almost exactly 500,000 times larger than what this protein molecule actually looks like. And so what does it look like? It looks kind of like a blob, right? That's what everyone always says. Why is it exciting to know what the structure of a molecule actually is? And so I'm going to pass this guy around, and so I'm going to put in this right orientation. There's a little orange mark here. This is actually the business end of this molecule. So this is a kinase, and so this is a molecule that turns signaling pathways on and off. It's something that, like, it's a molecular switch. It's something that, like, if this is active, You grow. If it's not active, it's off. And there's this very little pocket right here that's highlighted with this little orange ball. And I'll pass this around. People can look at that orange pocket. So maybe just, as you pass it around, show people that orange pocket. So it's this guy right there. So, and you look at the features of that protein. Essentially, it has these little grooves. It has little tiny pockets that are all around it. And if you can know the shape of those pockets, you can start developing a molecule that can fit into those different pockets. So when people ask, what do you actually do every day? That's really one of the main focuses of my lab, is that we try and take these proteins of extreme interest and we try and find out exactly what they look like. Now, the tricky bit is that, of course, that's not what a protein in your body actually behaves like. It's not like a solid white glob. It's a very dynamic structure. So it's always moving, it's always changing its shape. So we want to get kind of a static picture of what it looks like, but then we also want to know those like molecular motions and how it changes its wiggling and shaping. And so what we've been doing, and probably what we're most well known for, is we've developed in combination with two techniques. The first is a technique known as cryo-electron microscopy. It was what was given the Nobel Prize for in 2017. That's what I did a lot of my training in when I was in Cambridge. And so that's a technique that allows us to determine that kind of analysis, which is the structure of a molecule and what it looks like. And then the second piece of technology is something known as, I'll call it structural mass spectrometry, and that allows us to look at the dynamic wiggling of the protein, so at a very simplistic level. And so what we've been doing, and where we're making great strides, is we now know with high confidence, when I say this, that PA3K enzyme is mutated, and it's mutated in roughly 3 different positions, we know exactly the conformational changes, the shape changes that occur in that molecule. And so now what we're doing is we're using a wide variety of different medicinal chemistry approaches to actually design molecules that are specifically targeting that mutant version. And those first molecules have now come through. They're in clinical development. So that's something that my team has been involved with, is that we now have identified these molecules that really are mutant specific, and that hopefully those will be entering phase 1. And when I say my lab did that, of course, we did that with a team of about 200 other people. So we, of course, did the structural work. There's a great assembly of academics and industrial chemists who have all been working together on that team. But really understanding that basic science portion of how these molecules look, how these mutations change them, that's how you can start going towards the next level of drug development. Okay, I am going to switch to my second kind of point here, is I want to talk a little bit about other solutions that are coming through in drug discovery. So that was an example of how really it's called of, so we think about cancer. The first generation of cancer therapeutics were chemotherapies. Chemotherapies is you have every cell that divides rapidly, you kill all of them, right? It's great at killing the cancer. It's really hard as a side effect. It's something that if you've known someone who's gone through it or if you've gone through it yourself, you know the massive set of side effects that come through. The second kind of development in drug discovery that we've kind of talked about a little bit is that you identify the molecule of interest and what's known as a targeted therapy. So if it's PA3K, first generation, a targeted therapy was you turn that molecule off, but you turn off all of them, not mutant or wild type, right? And then you get all of the possible complications and toxicities, side effects from there. Where we are in kind of generation 2 of targeted therapies is we're now going towards how do you make molecules that are specific only for the mutant versions. And that's where these kind of different structural biology and biochemical approaches really can start to understand how these things work. Now, I want to talk about some other kind of approaches that also might be super exciting, and sticking with this PA3K example. Going back to those molecules that turned off all the PI3 kinases, when you had this hyperinsulin effect, can anyone think of another way that maybe you could still use that drug, but that would be combining it with something else? If you think about insulin in the body. Is there any other way we can control insulin? Diet, yeah. So there's actually multiple ways. There's one is that we can use diet. So now, like the actual generation of compounds is being given with a pharmaceutically prescribed diet. So essentially a diet where you try and minimize any insulin spikes. So you essentially completely remove sugar. Now, I will say it's really important that that is being prescribed in the context of the drug. There's actually some cancers that respond worse with just a pure ketogenic diet. So, you know, it's a combination of those two together. So I think that's another direction on where kind of drug discovery is going. It's actually that you start combining metabolic changes, so dietary changes, along with giving the drug, especially as you learn more and more about how these things work. The other thing you can do is you can start giving molecules with two different purposes. So there's multiple drugs now that also work for blood sugar. So in these kidney transporters, they have drugs that you can get, SGL2 inhibitors. People might have heard of those before. Those also essentially increase secretion of glucose in your kidneys, so through your urine. So it's a way that you can decrease blood sugar through a second approach. So you could give that same PI3K inhibitor, but now you also give another molecule where you secrete that blood sugar out, another way to take that insulin spike down and decrease the signaling through that. So that's kind of, you know, the aspects that are really pushing this next level of discovery is A, making better molecules to target the molecule of interest, B, figuring out how you can minimize the side effects by treating with other molecules. And then I do want to throw in this one kind of strange idea that maybe people have heard of just because of local news. What's probably the protein that most people have heard of in the audience in the last 2 years? Spike protein. Everyone's heard of the spike protein. This is the one time that now it's a protein, and probably most of you have seen a picture of the spike protein. It was on the front cover of the New York Times. I'm actually good friends with the person who solved that cryo-EM structure. I'm very jealous that that's kind of the top tier of the field you can get. But this is probably the most informed anyone in the world is of structural biology. But that spike protein, there's actually some really cool molecules people are using. And so what's the molecules we have heard, other than the spike protein, most about in the last 2 years that we make? When we get infected by a virus, what does our body produce? Antibodies. Once again, everyone's heard of antibodies. This is great. This is like the one positive of the pandemic is that people now have a better understanding on how these things work. Now, human antibodies are actually pretty complicated molecules. They're two chains. They have a lot of intricate bonds in between them. They're really hard to make. You can't just easily, so they've made monoclonal antibodies, and I'm sure people have heard of those. Regeneron has the one that's the most famous, where they actually generated antibodies that were specific against the spike. And I promise this is going to relate back to PI3 kinase in a second, but I at least have to introduce the concept of antibodies here. There actually turns out evolution can do very cool and strange things. If you look across all organisms that have antibodies, there's actually a surprising diversity in how different animals generate them. So antibodies start roughly around bony fishes. That's when you start getting an adaptive immune system. And two specific set classes of animals actually have really, really intriguing antibodies with cool properties. They're camelids, so it's camels, it's llamas, and it's sharks. All of those have these very small single-chain antibodies. Now, why is that something that I'm talking about here when I'm talking about COVID and I'm talking about PI3K? There actually are antibodies that were generated in a llama that are being used as a nasal spray drug candidate for SARS. The reason that's true is because you can't make our antibodies easily. They're really hard to make. You can, if it's a high enough target. These antibodies from a llama, you can make in any run-of-the-mill cell. My lab can make them no problem. Making 1 million antibodies, we can make them, but it's a lot. It costs us probably 100 times the scale. So now what they actually do is they can give these single-chain small antibodies from a llama, and they can inject that in a nasal spray, and it is a way you can protect yourself from COVID. That's something that's probably going to be clinically approved and might work. Why is this something that I'm bringing up in the context of the PI3K? We did a project where we actually took our wild-type molecule and our mutant molecule, and we injected into a llama the mutant version of that enzyme. So we purified that protein machine in large yield, and we actually put that in a syringe and injected it directly into the llama. Everyone says, is that a little bit nuts? And it kind of is a little bit nuts. And so what you do then is you take that llama. This doesn't hurt the llama at all. The llama probably has a maybe minor irritation at the site of the injection, but it doesn't hurt the llama. We'll make sure that for animal safety that this llama was totally happy. He lives in a beautiful farm in Belgium. This was probably like, the llama had a great day. It was still okay. And what you can do is then, our colleagues over in Belgium, they take the blood cells of that llama, and they use a variety of genetic approaches that they essentially can create a library of every antibody-producing cell in that llama. Same way we have 10 to the 12 different cells that make antibodies. That llama has 10 to the 12 different cells that make these cool single-chain antibodies. And so they screened all of those. We gave them more protein. They could look for binding. We found about 100 different unique antibodies that had never been identified before that bound to this molecule. What was cool about this is we found some of these molecules that specifically turned off only the mutant version and had no effect on the wild type version. So there's a lot of that's not quite ready for mass discovery of drug discovery, but it just shows kind of the cool kind of unique technologies that's coming there. But for surface proteins, so proteins exposed on the surface that you could actually directly inject this into someone, it's actually a technology that already can be approved for going towards drugs. So I think I've still got about, I'm just making sure my time, so 30 minutes in. Does anyone want to ask any questions now, and I can go into kind of the second phase of the talk, or people just want to do questions all at the end? Yeah, shoot.
What's your connection and your work with big pharma?
Okay, all right, loaded question. All right, so I do not take any research funds from big pharma. So I'm not a shill. I occasionally have worked with them. So we've collaborated. So I've received materials from them, and we've used that in our research, and I assist them, but we don't get paid for that in our actual research. So I will say that, and also, do you have a distinction between big pharma and small pharma? Because you do have a big distinction. Okay, so majority of our work is what I'd call in the biotech sphere. So I'd say the majority of companies that I collaborate with are about 500 people, and are not, they're going for one target. We've done some stuff with Novartis, which is a large-scale pharma, but we do not, none of the money that comes into my lab is coming from a pharmaceutical company. Yep, shoot.
How do you get protein structural information from mass spec?
Okay, great. Do you have a background in, how much detail do you want on this?
I'm a chemist.
Oh, perfect, okay. So I can explain this in a chemistry way. If people's eyes start glazing over, I promise this will be short. So we do a technique called the hydrogen deuterium exchange. And so essentially what you do is you expose that protein to a deuterated solution. And then you're a chemist, so you'll get, at the amide position, every amide can exchange with deuterium. And that amide exchange is dependent on the structure. So it's dependent on the actual hydrogen bonds that hold the protein together. And so essentially we use that as a molecular ruler, as that the dynamic ones exchange very rapidly. And then what we can do is we can shift it into a state where we turn off the hydrogen exchange, we chop the protein up into little pieces, we shoot those pieces into a mass spec, and we measure them. Everyone got that, right? Yep. Yeah. Okay, great. Any more questions before we move to kind of the next section? All right, perfect. So I might do a slight left turn. So that was kind of in the big pharma portion of my work. So that's something that's very highly applied. And there definitely are some issues in terms of how drug development happens at a big pharma scale. I want to talk about the one other aspect of drug discovery my lab works on. So we've been working with Medicines for Malarial Ventures, which is essentially a public, what's the best way of describing it? Essentially is a nonprofit that their entire goal is developing drugs for malaria. And that has to kind of work outside of the normal drug discovery scheme because there just isn't the funding to pay for anti-malarial drugs, but it's probably one of the greatest health challenges that are actually coming through. So most of that money comes from philanthropists. So I would say that the majority of those fundings have come from the Bill and Melinda Gates Foundation. They've been very generous with funds there. And that I think, maybe just to give an example of a different way of how drug discovery works. How that company actually, or how that MMV actually runs things, is that they created an consortium of probably 40 different labs, 40 different companies. We have no IP on any of the molecules that come through, and it's something that we're doing solely kind of as solving a general challenge. That's the one where I do receive research funds to do that work. And so that's an example of, once again, using those protein structural biology approaches and going through. Oh, sorry, I see you have hands up there.
Wonder what IP means?
Oh, sorry, intellectual property.
Thank
Yes, so when you've heard all sorts of issues about big pharma, many of the concerns are frequently over IP developed for some of those molecules and then not widely shared. Probably most well known for vaccine development right now is that some of the issues in IP there. So that's an example of a different partnership. In that case now, what we've actually been working on is we work on a very similar set of molecules. So PI3Ks in cancer, PI4Ks. There's very small differences between those enzymes, also something that we're experts in. And essentially now we're looking for a way to turn off only the malarial variant, the plasmodium, that's the actual parasite that causes the disease, and not turn off our version of that disease. So we don't want to have any toxicity in the person. Someone who's getting treated with this, you can't have any side effects whatsoever. So it's this exact same concept. In the previous concept, we're thinking about targeting a mutant form, it's only in a tumor, and not hitting the molecule, it's in every other cell in your body. Whereas in this example, we're thinking about only turning off the version that's in the parasite and not turning off the version that's in our body. We use exactly those same approaches, but now it's in a slightly different scheme where actually that's probably that's what I hope the actual future of drug development discovery is that these more kind of academic industrial partnerships that are driven by an interest in solving a true world health challenge with dramatically decreasing the amount of funds that come in. Thank you very much. Perfect. All right. So that's kind of, I only made about 30 or 40 minutes here for the initial part, but I'm guessing that this might have some more questions that could come through. I'm happy to talk on more things. Yep, go ahead.
Do you have a patent attorney on board to protect your IP? I mean, at what point the process does a patent attorney come in?
Very good questions. I view, so the work that we do, I'll repeat the question because then maybe not everyone can hear it. So do I have a patent attorney on board to look these things over? I do not, because I kind of view that I'm getting paid by the university. So I usually try and view all of the work that we're doing. It's funded by Canadian Institute of Health Research. We try and make sure everything we put out is open, right? We don't actually make the molecules. So this is the one thing, as the person who characterizes the structures of how these molecules and giving feedback on design, I don't have the interest in patenting it myself. I try and get that information out to as many people as possible, and then they can use that information to go forward. Now, I think there's an aspect maybe we could, but I think that working in an academic environment, I have the feeling that we a little bit, when we receive federal research funding, should be distributing that information as widely as humanly possible. Any other questions? As a professor, I can talk on any of these subjects forever. So if you want anything you want me to expand on, I'm also happy to.
So do you feel that the world of research in the areas you're interested in has changed because of COVID?
There have been positive developments. Oh, sorry, I'll repeat the question. Do I think that there has been, did you say positive or you say differences? Just changes. Have there been changes generically, good or bad, in the research environment due to COVID? I would say, I'm going to put on my glass half full view first, but I think scientific literacy is available more than it's ever been. I'm not sure the uptake is fully there. So I think more people understand the challenges and understand what an antibody is and understand those things. There's been a huge push in biotech doing new and different things because of really having to push under the time crunches of COVID. In the negative, so I didn't mention at all, like a future of drug discovery is definitely going to be vaccines, because I was trying to avoid controversy possibly a little bit. And so, for sure, everything that like drug targets, these RNA vaccines are going to be hugely impactful. And so I think that's probably at the day-to-day level, we're going to have anti-cancer vaccines coming that are based on that RNA technology probably within the next 10 years. And that was sped up massively by it. I have some concerns over its uptake, just in the politicization of it. So that's the glass half empty, is that maybe trust of science is a little bit lower than I would currently want it to be from the current last 2 years.
There's been some news about generic flu vaccine coming out of this research, but through COVID research. Do you have any comment on that?
Yeah, are you talking about like the one out of University of Washington, the one from the like engineered? Yeah, so that's like the nanoparticle. So that's definitely a development. So I think with those, because all of a sudden all this money went into vaccine development. I think that'll have payoffs in a lot of different aspects because they've really, at the nano scale, because that's essentially they make these very kind of small little machines that they've dotted that with the surface of every possible flu strain. So when we get a flu vaccine right now, they're usually, and I'm not an expert on the flu vaccine, so I should be very careful in saying that this is my current understanding, is that essentially you're only getting a small set of the strains. You actually can't get a broad spectrum. The new technology, what they're going for is that they have this kind of nanoparticle and that they dot that surface of the nanoparticle with many more strains than you actually could put through there. And so when you get that flu vaccine, your propensity to be protected is much higher because you're more likely to be exposed to the actual flu strain that's out in the circulating environment. So I think that is a positive development that's come from the COVID pandemic. Yeah, Scott.
John, are you done? Because I should be running back.
Oh, sorry. Yes, I am done. I think at this point I can go. Yeah. Yeah. I can also expand on a given area because I was planning to go kind of 30 minutes and then 30 minutes for questions.
Awesome. Well, great job, John. I feel a lot smarter. I was not expecting to learn about a happy Belgian llama tonight, so I'll add that to my list of new facts. So any further questions for John? I think he's brought up all sorts of super interesting points. Yeah, over here, please.
There's probably a fair bit of mRNA floating around in this room. Is it possible that, or should we trust them blindly in the future, or not?
There's probably a lot of mRNA floating around in the room. Should we be trusting the manufacturers of the stuff in the future?
Yes, I would say yes. So, I mean, the thing is, you got to remember all these things. You want to be skeptical at the beginning when there's no data, right? So that would be my view of big pharma is they're not evil, but they're not saints either, right? So there's a profit-driven motive. Now, my view as a scientist is that you have to follow the data. I think where big pharma has gone wrong is that it's been trying to overprescribe a given drug in a condition that drug doesn't have a benefit for. I don't think that for the mRNA vaccine that wasn't the case, and the safety data that's out there is the most probably well studied of any molecule that's ever gone into humans ever. We have more safety data on that than literally anything that people have ever injected to them. There's no comparable data set of the number of people who've had it. So that would be my viewpoint. Now, when they modify the mRNA vaccines for a different downstream role, they'll have to go through some similar safety testing downstream. But, trust the data that comes out of the safety testing and trust the person who knows how to interpret that safety testing, is what I'd say.
Further questions? Yes.
Where would you see your work going in 5, 10 years' time? What would you dream of?
So, John, what is the dream result for your lab in the next 5 to 10 years?
Okay, so this is what I was going to possibly bring up as the next section. So maybe I'll use this as a jumping point a little bit here, is what's really taking off is artificial intelligence-driven methods. So as I said, my lab really looks at the structures of molecules. The real groundbreaking discovery of the last year has been the fact that we pretty much can predict the structure of everything. There's no protein molecule that exists in nature. If we have a genetic sequence for it, we can guess what it looks like. Now, there's limitations. We can't at this phase predict how a molecule would bind to it. We can't predict how antibodies would bind to it. So I'd say for my lab, this has been like the most exciting year we've ever had because we've had the targets that we've been looking for forever, and we finally can see what they look like, right? So something that used to take us 2 years and maybe 10 people of man-person time to go after, you can now pull up on a computer and pull it up in a day. So that's by far the most exciting. Then going into the idea of how we can look at the dynamics of how that change in different mutants. That's really like what our lab does a lot of the time. Now, where I think it's going in 5 years is I think 5 years, AI might have the chance that it actually can crack that idea of actually looking for a small molecule that binds to those protein structures, right? So you could train, in theory, an AI algorithm that you know that this is, this target, predict every possible chemical structure it can bind. Can you find something that would only bind to this mutated form and not the other? That's in development. It's not there yet. That would be the pipe dream, at least in terms of like the work that I do, and it would speed up what we could do exponentially.
So John, this may be dating me a little bit, but a long time ago, when you had spare computer power, you could install a program called Folding at Home rather than mine cryptocurrency with your computer power. So your program basically is just a much, much faster version of what that distributed software was trying to do.
Yeah, so for that specific example, Folded at Home actually was using human intuition combined with chemical first principles to try and fold a protein. And that actually, for a long time, was the best thing that was there. These were completely non-trained chemists, but it was essentially a program anyone could install in their house. And it was like a game you could play. It was kind of a puzzle folding game. You try and fold this protein the way you think it should, and little things would light up and say, is this good or is this bad? That was actually probably one of the best computational ways you could do this. All right, so I was a little bit involved on the TT Rosetta stuff. So I'm gonna, once again, if I see too many eyes rolling over, I will cut this short. The way that these AI methods work is that essentially people like me, structural biologists, have generated about 30,000 protein structures. And those are all in a big database. And so there's essentially a well-known set of data for how a protein can fold into its given shape. What they did in this technique is essentially they then, anytime you have a protein of interest, if it's a protein that we have in our body, there's a chance that that might be conserved all the way back to yeast, right? Just through evolution. So what they did is they could actually take a sequence alignment, and so that's where you take the genetic sequence, so the actual amino acids that make up our protein are made out of nucleotides, so this is the DNA to protein, and they could take those 500 to 1,000 to 10,000 different genetic sequences, and then you look at coevolution of how that works. You combine that with template modeling, and you can create an artificial intelligence system that's all happening in a black box. It's a little bit like how AI methods work. You essentially have training sets. You train it with these proteins. You train it with these sequences. And essentially, they kept refining this algorithm until they got something that pretty much was completely indistinguishable from experimentally solved structures. So that's the story of how that happened. And even to a person like me who's spent 20 years of their life thinking almost exclusively about this, it's sometimes hard to wrap my head around. So it's definitely complicated, but the advantage of computer science integrating into the biochemical sciences field.
So is this similar at all to what they did with Deep Blue and teaching it to play chess?
Yeah, so DeepMind is actually the name of the company that did AlphaFold. Still from Google. It was essentially their level of complexity up from Deep Blue. Right. So DeepMind is the actual company at Google that did this work. Once again, Google did that all for free. They released that out. So, I welcome our corporate overlords possibly, but that's one example of a company that maybe you can say of the negative things. That was one very good positive thing they've done to the scientific community is they just dropped all of that data for free. They didn't keep that within a pharmaceutical environment.
Other questions? Yes, please.
Yeah, happy to talk.
So John, can you dig a little bit deeper into the various phases of trials, from animal trials to the different phases of human trials and what they all mean?
Yeah, so historically, and this is pretty accurate for still how majority of clinical trials work, is that essentially they have some molecule that they hit some target. most of the time now they actually have an identity of whatever protein or small molecule it's targeting. They then have to test in animals that when they give that molecule, does it turn off whatever they want to turn off, so kind of an efficacy. And they also have to check for its general toxicity. So does that molecule cause a systemic infection? And they usually kind of scale up. That's predominantly done in mice, but mice are quite different from us. Depending on the thing, they'll then go into a higher organism from there. And then once they're convinced that this is a molecule that's safe to give to animals, that's when it enters human testing. And so there's 3 phases of drug discovery for drugs. Phase 1 doesn't actually look, does the molecule treat the condition? It just looks, is that molecule safe? So if you're ever enrolled in a phase 1 trial, all they're looking for is we give you this compound, and they're just checking for a wide variety of side effects. They check your blood, they check, you have a questionnaire, you're going through all possible sensations you have, and they give that with a placebo, and they give it with the molecule, right? And so they can get from that initial phase 1, is this a molecule that is safe for the person to take? At that stage, then they want to enter phase 2. So so phase 1 is a relatively small set of people. Maybe you're talking at most 100 people, but sometimes kind of 10 to 50. Phase 2, you're also in kind of a small set of people, but now they're looking for efficacy. So you're actually looking, does this do what I want to do? So if it's for cancer, do they see regression of the tumor? Do they see progression-free survival? And if that has a very obvious positive result, they end that trial early and they institute what's called a phase 3 trial. Phase 3 trial is taking that phase 2 trial and scaling it up to a much broader population. Now, that's where the majority, it's about where 50% of the cost in drug discovery goes. So it's a lot of it is in the basic discovery. Where the money gets spent in is that many, many, many drugs fail at every one of those stages. So many examples of drugs that people were, killed every disease in a mouse, but has had no effect whatsoever in a human. So at every one of those phases, molecules fall off, but all that cost goes in. And so every time a drug succeeds, it's paying for that whole set of molecules everywhere else. Yep.
So then a person who's being considered for a clinical trial should have the opportunity to know if this is a phase 1 or a phase 2, like where they are along the progression of this trial.
So someone signing up for a trial does
Might be signing up for a trial.
does get told what phase they're at?
Ooh, for phase 2, phase 3, and I will say that the 100% is not an MD. So this is definitely a good question I can ask, because I do know people who run these kind of things. I do believe if it's phase 2 or phase 3. Phase 1, you're just, essentially that's for someone who's volunteered to essentially take a drug as that they're warned of this is safety. They're paid for it, right? So phase 1, you're essentially paid as a little bit of human guinea pig of, we will pay you to check this. They're as sure as they can be that it's safe from the animal model, but there's still a little bit of risk in that phase 1. There's dose escalation in that portion. Phase 2, phase 3, that you're in for this disease being treated with this molecule. You don't know if you're in the placebo arm or if you're in the control arm or the treatment arm. That they can't, and the doctor doesn't know that either. But they do tell you if it's phase 2 or phase 3. I believe, but once again, that's not someone who actually runs that. So current understanding of how that works is that's the way that phase 2 and phase 3 works.
So what proportion of drugs actually make it all the way through?
Less than 1%. So a molecule coming out of pre-clinical, so before it goes into phase, so they think they have a molecule that's going to go somewhere, it's less than 1%. And it's kind of an equal proportion die at every stage.
Do you think AI will increase that percentage?
Okay, glass half full. I'm hopeful, yes. But the issue, so if we go back to what I was talking about with AI in terms of a given target, AI wouldn't have predicted systemic feedback. A lot of the times where we probably need to be thinking of, at least for the systems I'm studying, you need to think about the effect of that when you hit every given molecule in the whole human body, right? Turning off a system in your liver versus turning off a system in the tumor versus turning off the system in your muscle will have a very different effect. Now, could you train an AI to eventually account for those issues? Possibly, but that's going to be the longer term. Right now, AI is really good for I've got molecule X that I'm pretty sure is important. It's going to speed up the generation of the molecule for that exponentially, right? So that immediately will take down cost because hopefully a lot of that work will no longer have to be done. Going to the phase 1, phase 2, phase 3, sometimes that's unexpected effects that I don't think there's a good training set for an AI yet.
Yes, please.
You've mentioned a couple of times the concept of folding, and your model of that molecule looks rather odd in its form. But I'm assuming that that's not what the thing looks like if you were to stretch it out. Is that what folding is?
Yeah, can you tell us a bit more about how folding works?
Yes, I'm trying to think of the best way to describe it. So I'll go from first principles first. So essentially, every time a protein gets made in your body, it's getting made at the ribosome. Ribosome is the big protein factory that's in your cell. And what's being happened is that protein spit out is all that chain of amino acids. And so what's happening is as the molecule's being made, it starts folding into its actual shape, right? And so many times this is assisted by a variety of other machines in the cell known as chaperones, and those work together. And essentially, for most proteins, their shape is encoded solely by their sequence. So if you know, that's how these AI methods can work, is that they may need a chaperone or some other help to get there, but essentially, chemically in the matter of the molecule, it encodes its final shape. Does that make sense? So then, what was the follow-up question on this in terms of the folding and?
So how does the folding itself happen? Is it just chemical bonds snapping into position? Is that the idea?
Okay, yeah, so depends on the system. So for some proteins that literally just those weak electrostatic and hydrophobic and all of the chemical bonds that Scott will be much more knowledgeable on than I am as the chemist. But essentially there's all of these weak interactions and exclusion of water, all these things. As the molecule comes, it just snaps into place, right? So this molecule here, this is not the way you usually look at a protein. You probably usually see them as like ribbons and diagrams, all those things. Those ribbons and swirls, if you've seen those in different publications, that's essentially showing some of those interactions that are occurring between the molecule. For big molecules, you do actually need sometimes a second set of molecules known as chaperones. And those are molecules that actually bind and stabilize around it, and they help with the folding process. So many diseases, so many neurodegenerative diseases actually are from problems in protein folding. So Alzheimer's disease, ALS, Parkinson's, those are actually diseases of protein folding where there's issues where the protein folding is disrupted. In a healthy person, in between the sequence itself and chaperones, they work together to make sure every protein is properly folded.
Yes, please.
I took Bio 101 a very long time ago, so this might seem like a really basic question. I really am curious why it's always, many times I read stuff it's about proteins only in the cell. Are there other parts of the cell that are as important as a protein in doing some of the kind of research that you're doing?
John, what are the other important parts of a cell apart from the proteins?
So that's a wonderful question, and it's going to let me bring up my favorite molecules. So there's lots of things that are important. So DNA is our blueprint, right? That's probably one everyone's heard of. RNA got the rap as the molecule that's the intermediate between proteins and DNA, but actually is really important on its own. RNA is also a molecule that can do all sorts of cool things in the body, and actually can almost behave like a protein in some systems, right? So if you think about the molecular workhorses, like if you could imagine a cell and you could zoom in really, really close, probably proteins and RNA are doing a majority of the work. The molecules I'm going to argue for that are understudied that are also really cool is that there's many of these lipid molecules, so like the actual constituents of all the little compartments of the cell that are changing at all time. Now, most of the time, it's an interplay in between how the proteins are changing some of those molecules, and they're changing metabolites, so different sugar derivatives, there's essentially a complicated interplay. Proteins are always involved, but they can sometimes be like the conductor, and then the instruments are the individual molecules that are changing as they're essentially manipulated by those proteins and altering them. Proteins are the ones that we've had an easier time to study because they're more experimentally tractable. A massive growth industry is now figuring out all of the molecules that those proteins are actually changing and seeing how those also affect downstream signals. Sorry, was that a Biochem 101 answer, or was that maybe too much? Okay.
So we've moved from the genome to the proteome. Does anyone have any final questions?
Yep.
Yeah, so are all new drugs based on this molecular protein kind of modeling and synthesization? Or are there still drugs being developed that are kind of where they take a naturally occurring compound or molecule in nature and then just refine it or isolate it, and it already fits the bill for whatever needs to be done?
So John, do you have any idea about the proportion of drug discovery that involves these more modern methods, or do the more traditional methods to do with isolating natural products and things still play a big role in development of new drugs?
So there is a massive set of drugs that are currently in use. They've been, so aspirin. Aspirin is a classic example. So my PhD, other than the snake venom, I worked on some of the aspects of one of the molecules that aspirin in theory regulates. We don't really understand how aspirin works. It covalently modifies a bunch of stuff in your body. It turns off pain signaling. But in terms of an exact target, it's still a little bit undefined. Probably that's the drug that the majority of people have had over, if you looked over the longest period of time, that's probably the most, doesn't even have to be prescribed, but most people have taken. So there are plenty of examples still of molecules that we take that we know have a specific function that we don't know exactly how that works. That being said, new molecules going forward, so there's still a lot of work in natural product discovery. So, people go and they look in soil samples, they look in seawater samples, and they look for molecules that bacteria are secreting that turn things off. There have been some unbelievably cool molecules that have been discovered that way. I'd say most of the time, I don't want to say most, greater than 60 or 70% of the time, so I guess that is most, people know what that target is now. So there's been a huge amount of work going into target discovery, and that's advanced so far now that when people pull out some new cool looking molecule from a microbial source, the technology is advanced that you can identify the target of that a lot easier than you could 20 years ago. And so almost everything going forward seems to have, as part of it, target driven into it, that you actually want to know what that molecule is doing.
All right, if there are no further questions, we're past the 7 o'clock point, so I'm going to wrap it up there. John will be available afterwards to answer any questions if you want a one-on-one with him. But John, thank you so much.
Thanks, everyone.